Good morning, everyone, and welcome to Grand Rounds. A couple of announcemements first. One is that Sarah Canetta received an RSAD, or Brain Behavior Research Foundation, Young Investigator Grant for a project on activity-dependent competition between long-range inputs to prefrontal cortex during adolescence. And Susan Vaughn was accepted as a candidate, or as a participant in the Columbia University Leadership and Management course for women faculty. Congratulations, Susan. Some people are gluttons for punishment, but I'm sure it'll be for good purpose. Next week, our Grand Round speaker is going to be Christina Alberini from NYU, who will be speaking on the biological bases of infantile amnesia. Infantile amnesia, a critical period in learning and memory. And this is the Roger McKinnon Memorial Grand Rounds. I think hopefully people have a sense of the fact that what our department has been trying to do is push the envelope, so to speak, with respect to the kinds of research, clinical initiatives, training experiences that we provide. And this is reflected to a considerable degree in the strategic plan that circulated in draft form and will come out in final form in March at the latest. But one of the ways that we're doing it is sort of exemplified in the Grand Rounds today, which is to establish collaborations with disciplines that are not traditional for academic psychiatry departments. And in this case, it's engineering. So Paul Sajda is our speaker today. And Paul is a graduate of MIT, where his bachelor's thesis was on machine implementation of a human motor task, the yo-yo robot, and it was the best undergraduate thesis in electrical engineering. He then went to Penn for his advanced education in bioengineering and received his Ph.D. there. His thesis was reverse engineering of intermediate level vision, surface segmentation, and depth from occlusion. And this also received the Best Doctoral Thesis Award of Bioengineering. And then he was recruited to Columbia in the year 2000, where he became director of the Laboratory for Intelligent Imaging and Neural Engineering, and also has ascended to the rank of professor in biomedical engineering, electrical engineering, and radiology. And just sort of mentioning a couple of his professional scientific organization memberships will tell you how he has placed himself at the interface between neuroscience and engineering. So he's a member of the SFN, Society for Neuroscience, which you don't find too many engineers, a member of the American Institute for Medical Biological Engineering and the American Association for the Advancement of Science. So he is an engineer who's sort of already acquainted himself with various life science disciplines in a way that engineering can inform. His area of research has been, I guess, I couldn't characterize it in my own words, so I'll use his, using principles of reverse neuroengineering to characterize the cortical networks underlying perceptual and cognitive processes, such as rapid decision-making in the human brain. His lab uses basic and applied neuroscience research techniques with an emphasis on non-invasive multimodal neural imaging, visual perception, brain-computer interfaces, application of machine learning to analysis of neural data, and computational modeling of large neural systems. So I can't tell you exactly what all that is, but it sounds really good, and hopefully he'll explain it in the context of his talk. So please join me in welcoming Dr. Paul Sajda.
So thanks very much for the invitation to speak to you. So what I want to do is kind of go through some of the work that we've been doing over the last decade or so, fusing simultaneously acquired EEG and fMRI to really get a better idea of the spatio and temporal dynamics underlying what are relatively simple cognitive processes, rapid decision-making, but shed some light, I believe, on at least more complex cognition, and ultimately have some value for perhaps understanding different aspects of mental illness in which dysfunctional decision-making might occur. So just financial conflicts of interest. One thing that Jeffrey didn't mention is I actually, between my PhD and coming to Columbia, I was in industry. I kind of got a little bit of the bug of, in addition to doing science and engineering and academia, being able to take some of what we've done and kind of be involved in different spin-offs and companies like that. So I have some potential disclosures to make here. So what I want to start off with is kind of, so where we are in being able to measure activity in the brain. And this is taken from a paper by Terry Sinofsky and others, and it shows kind of the landscape of spatiotemporal resolution that was first around 1988, so how well covered was this space, our ability to record in this space brain activity across space and time, and where we were in 2014, okay? Now, 2014 doesn't seem that long ago. Actually, it's, 4 years ago. But it turns out, because of things like the Brain Initiative, right, this space has been filled up even more, right? And so the Brain Initiative itself has focused quite a bit on trying to improve our ability to capture very high temporal resolution across entire brains. Now, one thing, however, that I think also is true is that the technology development has been greatly focused on basically technologies that we would consider invasive, or at least nowhere near a point where we can employ them in human imaging, particularly, let's say, in psychiatry. So needless to say, even though there's a lot of excitement in terms of how this space has been tessellated, we're still kind of up here in terms of our ability to look at the functioning human brain and measuring that across space and time. And kind of two of the workhorses of that in terms of measuring function in the brain, of course, are EEG and MEG and fMRI, right? And in fact, one of the things about these two modalities is that they actually don't intersect really in terms of the spatial and temporal resolution. You get, let's say, much better spatial resolution down to maybe layers, depending on the field of your MRI, with fMRI imaging, but the temporal resolution is rather coarse. You're on the order of seconds, at least with kind of conventional techniques. EEG and MEG, on the other hand, we can get much closer to resolutions that represent the underlying dynamics of neurons, on the order of milliseconds or tens of milliseconds. But because of the very nature of these being recordings that more or less are topographic, or at the very least are not true 3D imaging modalities, our ability to really localize across space is limited. And so one idea, and it wasn't my idea, years ago, was, well, could we come up with a modality that combines the two, that essentially allows us to fill in some of this space by collecting the two sets of data simultaneously. So sounds like a good idea, and in fact, there are a number of different reasons to think about acquiring these simultaneously from a scientific point of view. One actually has to do with just really understanding fMRI techniques that are really the workhorse of cognitive neuroscience and understanding brain function, particularly since they're measuring something indirect to neural activity, right? They're measuring hemodynamics. And so if we can, in a population of healthy individuals, human subjects, maybe doing different types of tasks, really measure the electrical activity via EEG and look at how that correlates or is related to the underlying hemodynamic changes, we might get a better idea of the relationship. And there's a lot of work that's being done on animal models and using invasive methods, but this might allow us to actually look at this in a way where we can really use non-invasive experiments. The other thing is obviously this idea of in the two spaces of spatial and temporal resolution, can we kind of leverage the spatial resolution in the fMRI together with the temporal resolution in the EEG to kind of create a construct, a representation that combines both. So there have been people, for instance, that have said, well, what if we use fMRI information, so the hemodynamic response after we do some type of modeling, and those locations where we find activations, we seed EEG source localization. So EEG source localization is kind of the method for taking all these measurements from the scalp and kind of solving an inverse problem to localize that activity in the brain. It's hard to do without some prior modeling, so why not use fMRI as this prior model? And you can also flip it around and try to use the actual ERPs, trial average ERPs, to inform the BOLD. So this it sounds like a really good idea, but in some cases, when we think about how we might do this particular type of fusion of the data, if we're going to actually do a whole fMRI analysis traditionally to find these blobs or localization, so that typically requires the entire experiment for analyzing, or we're going to construct trial average DRPs, once again averaging over trial. There might not be a lot of advantage of acquiring the two modalities at the same time, right? Because what I'm going to talk to you about in the next slide is the fact that there's no free lunch here. So acquiring these two modalities at the same time is going to result in signal loss. So you have to have really kind of a compelling reason to justify that signal loss. And what I'm going to argue is that the real justification, at least the way we see it, is this last reason to acquire these simultaneously. Namely, that there's variations on a trial-to-trial basis. So this is if you're having, let's say you have an event-related design, so you can defy things in trials. There's underlying relationships between the variations in the EEG that relate to latent states in the brain, shifting of attention, mind wandering, things that might be representative of decision evidence. And there's some fraction of that variation that's not just noise, but it's meaningful in terms of the underlying brain state. And we can combine that information with the hemodynamic activity to make inferences about these latent states where we wouldn't be able to make those inferences if we just looked at what the stimulus was or what the response was of the individual while they were doing a task. And I'm going to really spend most of the time today just going through a number of different examples where we demonstrate the utility of coupling the trial-to-trial variability or the ongoing variability in a resting state task between the two modalities and how that allows us to make inferences about latent brain states.
Can I ask a question?
Sure.
Just clarification. When you say loss of signal strength, you mean loss of MRI signal strength?
Both. So I'm going to show you right now where you lose the signal strength, both in terms of the EEG and the fMRI. So the first thing that happens, right, is we're now going to combine these two modalities. This is actually a cartoon indicating we're looking top-down in an MR suite. So we've got our scanner over here, our person here, we've got our tech over here, and maybe an office area over here. So everything becomes more complicated because now we got to bring these two pieces of equipment together, right? And I don't mean just complicated because, you know, it's bigger pain to set up, but now we've got a lot more wires, right? So we've got wires in magnetic fields, and we all know that's a bad thing, right? I mean, so there's extra care we need to worry about in terms of setting this up. In addition, you know, depending on our experiment, we have different now environmental factors that we might have to take into consideration when we're thinking about designing, let's say, a task. So let's say we care about studying auditory perception or decision-making. Well, now, instead of doing this in a nice, closed, you know, anechoic chamber with EEG, now we've got, you know, some gradients pounding away. And so those might seem small, but there are all kinds of issues that we have to deal with. Really, the main challenge, and what Jeff's question was really getting at is, all right, so assuming we can deal with that, what are our problems in terms of signal to noise? So one problem has to do with how the wires and the sensors on the cap affect the homogeneity of the field that is in the MRI scanner. Okay, so those of you that know about MRI, you know that field inhomogeneity can cause what's bias fields, and the scanner tries to correct those bias fields dynamically. But if we put other things into the scanner that are metallic, what they can do is they can affect that bias field, and it's hard to correct. So here, this is an EPI image, so an echoplanar image for the functional scan, where you see this highlighting down here. That's where the wires are coming out of the head coil, okay? So, so here we get a hit on the signal to noise unless we're smart in terms of how we correct that bias field. So there are smart ways that we can do that. And in some cases, for instance, people never even take this into account. Everybody always talks about the artifacts in the EEG, but it turns out that there are a lot of systems. I'm not going to name names in terms of the commercial systems, but there are some commercial systems that love to have lots and lots of electrodes on the head, and these big, these tethers of wire coming out. If you ever look at those EPI images, it just looks almost like a binary image. Okay, so that's one loss in signal to noise. The other that everybody traditionally talks about is with the EEG. Okay, so I don't know how many people here are EEG folks, but this is a case where we have 49 channels across time. Those of you that know what EEG looks like, you'd say, this doesn't look like EEG, right? And in fact, it doesn't, largely because of a very strong artifact here, which is due to the gradients of the MR coming on and off, right? So what we have is somebody in this scanner with these wires, okay? These wires make loops naturally in terms of going back to the amplifier and being connected to ground. And the gradients through these loops induce currents, okay? So that's what you're seeing here. This is completely saturated by these gradient artifacts. Now, it looks almost like an impossible task, but the good thing is we know when those gradients are on. We actually synchronize the scanner clock to our EEG system, and we can use relatively simple signal processing to remove these gradients and get a signal that looks more like this. Okay, so right away, this looks a little bit more like EEG. It's still a bit noisy. But those of you that are also EEG aficionados or know a little bit about the challenges of acquiring EEG in a scanner, you can see that just about every second, there's this artifact that kind of sweeps. And in fact, this is the ECG at the bottom. It's kind of time-locked to the ECG, and that's called the ballistocardiogram. So this first artifact is due to the gradients firing, okay? This second artifact, the BCG, is due to the large static field. So you put somebody in the scanner, right? Hopefully they're alive when you do it. We're doing human neuroscience. We care about them breathing. Their blood is flowing. And so just with the little bit of pulsation of the electrodes on the skull, on the scalp, that little bit of pulsation in a 3 Tesla field induces these ballistocardiogram artifacts. So when you look at these, you say, well, it should be easier to remove than these guys because it's not quite as strong an artifact. But that's actually not the case. And the reason is that these artifacts essentially add to the system in a nonlinear way, because the way the scalp pulsates, its orientation of the little coils relative to the coils of wire relative to the field are all a little bit different. So we have to use more sophisticated methods for reducing this artifact. Now, in general, when all is said and done, we get about, let's say, 10% reduction in the SNR of our 2 modalities relative to acquiring them individually, right? So if you can really tolerate that, if that's okay, could be because it's just much easier to do the recordings together. You don't want to do it at 2 different times. But hopefully the reason is there's some kind of intelligent way you're going to combine the data, like I said, exploiting something about the trial-of-trial variability or the ongoing oscillatory information that you might see in the EEG during a task and coupling that to the bold. And in fact, what I'm going to really spend all my time on is talking about different ways we've fused this information together, particularly using different aspects of machine learning and data science. So the rest of my talk, the outline, goes through three different projects. I'll see if I have time for all three, and talks about kind of how we can make inferences within regard to processes underlying rapid decision-making in the human brain using multimodal imaging. And then at the end, the last slide, talk a little bit about some of the work that we're doing in collaboration with some folks here in psychiatry, as well as at other universities in psychiatry and clinical departments to kind of use this modality to consider how it gives us some advantage in characterizing mental illness. So those of you that know a little bit about rapid decision-making and cognitive neuroscience, you know that this has really been of interest, particularly in understanding coupling and the neural correlates of decision-making processes in the brain and behavior for a couple of decades. Mike Shadlin, who's now here in neuroscience, is one of the pioneers of this. He did work with Bill Newsom and others where they use monkeys and this classic random dot motion kinetogram where the idea was a monkey had to decide the direction that a fraction of these dots were moving coherently. So they could adjust what fraction of those dots are moving left or right. Monkey had to decide, and the fraction allowed them to kind of titrate the difficulty of the task. And since then, in addition to looking at non-human primates, this has been kind of a domain where people are particularly interested in characterizing decision-making in humans across not only, let's say, visual stimuli, but auditory stimuli, tactile stimuli, and so forth. In addition, I don't know about you, but pitchers and catchers are reporting soon. I'm a baseball fan, and this whole idea of, well, rapid decision-making, trying to understand, does monkey get a little juice reward if it sees something move to the left or to the right? Well, these guys get like $20 million a year to effectively make a decision in about 400 milliseconds, almost about the same amount of time. And so the notion of what is going on in the brain that allows us to very quickly evaluate a stimulus and decide how to act on it clearly has all types of applications in human behavior, in real-world human behavior. And in fact, there's lots of different books, Nobel Prize-winning work on this idea of how do we really decide very quickly. And so that's kind of what I want to focus on here. And in particular, talk a little bit about what's happening, kind of the themes in research and how we're going to kind of add to what's known about rapid decision-making in human beings by using multimodal imaging. So most experiments that we see, whether they're in non-human primates or human experiments, there's some stimulus that's corrupted by noise, and the individual has to make some kind of response. Eye movement, press a button, what have you. And so there's kind of this idea, this high-level model of what has to be going on in the brain, that the evidence in the sensory space has to be represented and encoded, has to be accumulated over space and time, has to be characterized in order to generate an action. So this makes it look, very feed-forward. But, the brain is a network, and it's unlikely that some feed-forward process really drives even these very rapid responses and decisions. So really what we're looking at is a set of processes and interactions that go from how the information is encoded to not just how an action is generated, but also how uncertainty is represented and perhaps used to affect the decision or the next decision, how something about the monitoring of performance might be used to update action plans and so forth. So the real challenge, the inferences we're trying to make are more kind of rich than that simple, the simple feed-forward scheme. In addition, many times when one is looking at this, we think about this as a discrete thing that happens. And so we have one one choice, then we move to the next one, and that next choice is independent of this choice, and all of our analysis is kind of set up to make those assumptions of independent, identically distributed events. And it turns out we know that what our state of mind is before we saw this stimulus likely contributes to how we evaluate that stimulus. And based on how we respond and any feedback we might get, that's going to affect what happens after we respond. And do we update our model? Do we change our preferences and so forth? So what I want to do is talk a little, and then the last thing is, what's really interesting about these types of tasks is I can give you the same stimulus, right? And I get variability on different trials. So I give you the same exact stimulus, maybe you're a little longer on one trial, or faster, you're faster response time, or you're incorrect, right? So that type of variability, uncertainty, we would try to, or what we're going to test is whether that's completely random. Why it doesn't make a lot of sense why the brain would just be random, or is it reflecting some other latent processes that are going on some trials, like, oh, my mind is wandering, or I'm more vigilant on this trial, and can we kind of make inferences using the methodologies of EEG and fMRI? Okay, so the first set of experiments I want to talk about, so Jordan Maraskin, Ph.D. student in my lab who graduated a couple years ago, he did some work where we were looking at this rapid decision-making task where subjects had to make choices between faces and cars. So these things were shown for 50 milliseconds. They had to respond as quickly as possible. And we could basically make the task more difficult by changing the phase coherence in the imagery. So it doesn't matter the specifics of it, but essentially it's modulating a fraction of the Fourier phase space. So this is an easier choice. This is a harder choice. When it's presented at 50 milliseconds, it's even clearer that this is a hard choice, and this is not that easy. Anyway, you can do the task and monitor the behavior, and you can see, not surprisingly, there's kind of this trade-off in accuracy and in response time, and in fact, for high stimulus evidence, so in these high phase coherence trials, subjects are more accurate and they respond faster, right? So nothing surprising here. Now, people have looked at these two sets of, or this experiment with EEG and fMRI separately, okay? And they found, for instance, that across time, so this is essentially an event-related potential from the onset of the stimulus to about 800 milliseconds after. This difference in, and this is over CZ, so right over the center of the head, basically a difference between the high stimulus evidence trials and the low stimulus evidence trials. So you see reflected across the whole trial some difference in the underlying activity in the EEG that reflects how much evidence there is for the stimulus. Same thing in the bold analysis. So if you do that just fMRI, you see areas like fusiform gyrus, not surprising because we're dealing with faces and objects. You also see part of dorsolateral prefrontal cortex, the anterior cingulate. But the point is, this is a snapshot across all time, and this is one point in space. Can we combine these in some reasonable way that we see something more of what's happening with all of these different activations as a function of moving from the onset of the stimulus to the response? So the first thing we're going to do is we're going to use some machine learning, and relatively simple machine learning, to try to tie these two modalities together. And so the way I want to start this is to think about, if we go back to the classic Shadlin experiment in monkeys, okay? So in that random dot experiment, what they were able to show, because they recorded with electrodes, that if you look at area MT, which represents motion, there's kind of a step function of activity that reflects the level of coherence in the moving dots. So more dots are moving coherently to the right or the left. You get a step and you get something high up here. If there are fewer moving dots, essentially it's a harder task, you get the blue step. And each of these individual ones are trials. Later on in the processing stream, in what lateral interparietal area, LIP, there's kind of an accumulation of this step function, an integration, and that integration is such that you get faster integration if there's more evidence in the stimulus, and you get slower integration if there's less evidence in the stimulus. So the key thing about this is at two different regions and at two different times, because if you'll know it early in the trial or early in the task, each one of these individual curves represent a different trial. There's separation between the high coherence and low coherence. Earlier here, that kind of goes away later in the trial. And here in region 2, it's not so clear here, but it's actually more clear later in the trial. Okay, so there's regional differentiation as well as time differentiation. And if we take that and within this data set, so try to separate apart, okay, are we looking at a trial that is high stimulus evidence or low stimulus evidence? And we build a classifier that puts down what's called a hyperplane, okay, essentially a decision boundary. We now think about this not in the context of these recordings in monkey, but in EEG. We have a space in EEG. So let's say we have 64 channels. So we have a 64-dimensional space. We learn this hyperplane, which I'm just showing as a line here for convenience. And this line is dividing all those trials in which the stimulus evidence was low versus high. Same thing happens here. Now this is a different hyperplane because it's actually weighting the sensors differently because the information is at a different region and at a different time. Now how are we going to use this information in a way that allows us to link it to the bold data? So far I've just processed the EEG data. Well, look at the relationship between the trials and the distance relative to this plane, right? So trial 4 has this distance y tau 1 4 over here, right? So we can think about this as this is something about the variability of this particular trial relative to this plane. And we can take this variability across all our red dots and create and use essentially that as a temporal tag. That tags all the trials at this time, okay, with a particular type of variability. This one has actually a different set of variability. You could see that trial 4, which is the same trial as here, later on has a different distance. These temporal tags now allow us to fuse the information in a way that we can combine the EEG and fMRI synergistically. And the way we do that is we take from the playbook of encoding models. Okay, so those of you, I don't know if you've, I think it was the beginning of last year maybe, there was on the cover of Nature, there's this beautiful picture of the brain and how semantics is represented all across the brain out of Jack Gallant's lab. So what Jack Gallant does to do his studies and make those kind of nice fancy pictures, he says, well, let me take a set of data. So I'm going to use images here. Let me take some images, and what I'm going to do is I'm going to calculate some features using computer vision, okay, right? So I'm going to be able to say, all right, so this is a image of a face. It's got some edges, so I can extract edge features. It might have a way I can describe a surface. I also have semantics, like it's a face. So all that information I take out, and I create this big vector that then I try to encode using the bold data. So I'm trying to estimate essentially a model that maps those features to bold data, and then I look to see how well I can capture those features relative to the bold data itself. So I fit this model, I try to predict the bold data, and I see how well I predict. So that's an encoding model, and from that, that's kind of a nice way to do a fusion of these features and the bold data, because you can do things like prediction, you can see how good the model fits, so forth. So our approach, instead of extracting these features from the image themselves, we use information from the EEG, from the brain, and those temporal tags as our features, okay? And those temporal tags, what that allows us to do is, whereas Jack's work allows us to basically label voxels by what their semantics are, So what voxels represent cars, and I can here it allows us to say, all right, what voxels actually are related to activity in the EEG at time 1, or tau 1, or tau 2, or tau 3. So we are now can kind of take those tags and use them to kind of show what voxels are covarying with the variability we see in the EEG.
Paul, can I
Sure.
ask a clarifying question? So you've got, a stimulus, which is your task, and you have a cognitive or behavioral response, so that you can measure, and that's not technically challenging. But then you're trying to combine these multiple modalities, and the reason that you are doing this is because, on one hand, you need the temporal specificity that EEG gives you, and you need the spatial specificity that the bold gives you.
Yes.
So you can see where the activity is coming from.
That's right. That's right. And I think probably this next set of results will make it clear kind of what you gain by doing this, particularly seeing activity that we wouldn't otherwise be able to see using either approach by themselves. So I won't spend a lot of time on this. I can go through and show how we kind of validate the model in terms of how well, by doing this type of modeling, it predicts the underlying hemodynamic response, or how well it predicts the actual response in the EEG. So, I can point people to papers about that, all that validation. But what comes out now is a set of maps where, instead of having one map before, like I had, let's say, here, which is across the entire trial, all time lumped together, now what I do is I get a cascade where I can pull apart activity at specific times within the trial and what voxels covary in terms of their hemodynamic response, okay? And so for this task, what we see, perhaps not surprisingly, is that the first significant voxels turn on at around 175 milliseconds. Maybe not surprising, given that we're looking at things related to object recognition, particularly faces, and we know that there's strong activity in the EEG at around 170. And then this cascade includes kind of a dorsal and ventral component. We see activity up through posterior areas, as well through the fusiform gyrus. The difference in the coloring just indicate on whether those voxels are more sensitive to high evidence trials. So the trials, it's really encoding something about a lot of evidence being in the stimulus, or not a lot of evidence being in the stimulus, okay? So yeah, you get a nice map like this, but what does it mean, right? So what does it tell us that we don't know from previous results? And of course, you can make fancy movies and all this, but let's first take a look at this a little bit more closely, and with respect to particular areas in the brain. So these are 8 different areas, and what I'm plotting here are these functions are actually the weights that the model learns, and wherever there's an asterisk, it means that that's a significant weight. So at that time, that there was significant activity at that voxel. And so the first thing to point out is we've got our red and blue, like I've shown before, but we also have green, okay? So what does this green actually mean? So if we take a case, let's say here, activity in the superior lateral occipital cortex, we have a time in which early in the trial, there's activity in one direction, okay, for this LOC, and later it's in the other direction. If we go back to our standard fMRI analysis, our GLM, we don't see these voxels activated. They're gone. And why is that? Well, because the way standard fMRI analysis is, it lumps all this together. So this positive and this negative cancel each other out. So you really miss activity in the bold imaging if you're doing these event-related designs with a general linear model, if those areas are coming on and off at different times. And you'll see there are a number of those. I mean, it's not just this area, it's left fusiform gyrus and so forth. So that's one thing. And we can also take this and kind of try to unwrap it and plot it in a way where we take the areas where we find significant activation and we kind of order them in a natural order from sensory processing to decision formation and decision monitoring. So this is just from the literature. And we look at the relative timing. So this is the timing at which these, there are voxels that are significant, and then this is essentially the response time and a kind of the variation around response time. And one thing that we found that we thought was super interesting was there's kind of this cascade where activity goes up to anterior cingulate. Then at the anterior cingulate, there's kind of a replay of this cascade during the response. So we thought to ourselves, well, what could this possibly be? It's not, it's definitely, it's not exactly the same cascade. It's definitely faster. It plays faster. And it's happening during the response time, which no one has ever reported before. So there had been work done in the cognitive neuroscience community talking about replay. Replay is actually really hot in the world of hippocampus. There's also some work in kind of long-term metacognition related to replay. And in fact, those of you that monitor all the crazy stuff that's happening in artificial intelligence and deep learning, the whole reason, or one of the big reasons these AlphaGo algorithms have been able to beat the masters is they use essentially a replay. This idea of they monitor the decisions that they were made and they kind of replay decisions that they had less confidence in. So we looked at this within the context of trying to better understand when this replay was occurring. And so we could go back and look at the behavioral data, okay, that for this task. And we used a cognitive or mathematical behavioral model to capture variables related to the behavior. This is called the drift diffusion model. It's a sequential sampling model. And not to get into all the details of this, but from this model, what we could do is we could construct a proxy of the confidence the individual had on every trial. Okay, and it's related to the response time and so forth. And from that, we could correlate the strength of that replay, okay, to or see if there was a correlation between the strength of that replay and the confidence. And what we found, so regardless of how we did the analysis, whether we locked to the response or the stimulus, is that if there was less reactivation, they were more confident. And this is regardless of whether they were looking at confidence in one trial type, the low stimulus trial type, or high stimulus trial type. So it wasn't just a result of there being two trial types with two huge differences in confidence, even within a trial type. And if there was more reactivation when the confidence was less, okay? So this seems to suggest that there's this replay effect going on each trial when somebody's making a decision. Now, why is that interesting? Well, when we think about these tasks, we often think, well, people aren't learning, they're not adapting, there's no reward, right? But one argument that has been coming out recently in the decision literature is, well, there's explicit reward and there's also implicit rewards. And implicit rewards might be just related to the confidence in the decision. And you might be using that, the brain might be using that to update their policies and its policy or model. And in fact, this offers a way where that confidence can be calculated on the fly by replaying that task through the same circuitry that were used to make the decision. So this is ongoing work. We're further exploring this by looking a little bit more closely at the task and redesigning the task to include a specific confidence measure. All right, so I'm running a little bit behind here. How much time do I have left? About, I want to have time for questions, so. I could do 10 more minutes, or, okay. All right, so I want to say something about at the beginning of the task and something at the end of the task. So we go into this task, and of course, what our state of mind is, or what we've learned even before we got into this experiment is going to affect on how we make this decision. So we look at, this is work by Tao Tu, a current graduate student in my class, and we look at essentially the same experiment, modified a little bit different ways. It's not so important here in terms of the timing. But what we're particularly interested in here is instead of differences in the level of stimulus information for the choice, like do I have a noisy stimulus or a relatively high fidelity stimulus, how are our choices influenced by the category? So here we've got 3 different categories: faces, cars, and houses, and we can look at response time and accuracy for faces versus non-face stimuli. And perhaps also not surprisingly, that we see that for face stimuli, regardless of the coherence level, subjects are faster, so their response time is less, and their accuracies are higher. We're better at recognizing these faces. And so one question is, why is that? What is it in the spatiotemporal dynamics that tells us something about what might be the underlying basis for this? And so once again, we use a set of, or an approach that uses trial-to-trial variability in the EEG and machine learning to now characterize differences that are not related to stimulus evidence, but are related to categories. So we train classifiers that characterize faces from non-faces. We can look at the efficacy of those classifiers. But the key thing is we take the variability, the trial-to-trial variability of those classifiers at 2 different times. Here we just lump that. We don't use it at 25 milliseconds. We actually just use 2 different times, early in the trial and late in the trial. And we look at encoding that within the bold data. And so what we find are 2 specific, what we call subnetworks or subsystems that clearly correlate with the variability for face and non-face classification in the EEG. We call it this early sensory system and this late sensory system. And from that, what we're in particular interested in is not what areas are different between, let's say, faces and non-faces, but the interactions between areas. So we use a method that's called multivariate dynamical system analysis. It's related to something called dynamical causal modeling. And the idea is that we can take the activity in the different areas that we observe in these 2 subsystems, so the red being the early, the green being the late, and model their interactions, okay? So these links here represent the interactions, particularly as they reflect a contrast of face activity is greater than house or face less than house. But really the key thing is the interactions are the key variable we're going to look at. And we're going to look at that with respect to behavior. And what we found was, this is kind of complicated, but the key thing to note is as you move up this axis, this is bias for making a face choice. So all else things being equal, you know, you have some image that has a lot of noise. The higher you up on this axis, the more likely you are to be biased towards faces. In both, if the alternative is houses or the alternative is cars, each one of these is a different person in the experiment. And this represents how much interaction there is in these networks between the late component and the early component subsystem. So here's a case, somebody with not a lot of interaction, okay? And what they would be doing is they'd fall somewhere here on these plots, okay? But if there's a lot of interaction, okay, then people fall up here. So it's almost like the more top-down interaction there is back to sensory system, the more likely they're going to be biased towards faces. And in fact, this kind of is consistent with a Bayesian interpretation that people have been looking at in terms of how do we make decisions, how important is bias in terms of predictive modeling and encoding. And if we analyze these networks, we're pretty much consistent with that idea. And in fact, what we're trying to do now is even look at some of these modulations in terms of the specificity of the different areas. So not just the gross connections between the 2 subsystems, but some finer specificity in those interactions. Yeah.
What are the sources of bias?
So I think in this case, right, it's very clear that we're naturally biased towards faces, right? So we're just better at it. We see faces in the clouds and things like that. I think a good question would be and a nice experiment would be to see if you could even introduce bias within a task and see these interactions form dynamically. I think also there might be some interesting kind of clinical conditions where bias might feed in, and we can kind of see, is this a potential marker for those types?
Yeah, excuse my ignorance, but I mean, is this the same kind of bias that like Malcolm Gladwell was referring to in Blink?
Yeah, kind of. I mean, it's a very low-level bias in the sense that we're dealing with.
But I mean, you're saying with regard to faces that, human beings may have sort of a natural biologic tendency to view certain things preferentially, as opposed to prejudices, which are.
Right, right. So that's kind of a higher level cognitive bias. This is much more like a perceptual bias. And yeah, I think one question is how things like societal-based biases versus perceptual bias, is there a common substrate in the brain, an interaction that that tells us something about the types of biases people form? Okay, so I'm just about out of time, but I don't want to just stop there because I want to say something about baseball. Okay, and so what about after the decision, okay? And so one thing that we did was we wanted to look, we did this whole study with baseball players because in addition to, doing studies with grad students and things like that, sometimes it's really cool to just see somebody is really good at a task, right? Like hitting a baseball. In fact, in particular, in baseball, one of the key things is, can you recognize this pitch that's coming at you at 90 miles an hour? You only have 250 milliseconds to actually start your swing. So it's a very rapid decision-making task. And one of the key things that really good players are able to do is they're able to guess which pitch is coming, okay? So you got a fastball, curveball, slider. They have different trajectories in space. And so if you have an idea of what is coming, you can naturally kind of adapt your swing on the fly to maximize contact. So we did this experiment where we had players and non-players do a go, no-go task that's modeled after how a baseball player solves this problem of identifying the pitch that's coming. And so there's this countdown bar where they're supposed to pay attention for either fastball, curveball, or slider on each trial. And then they see this pitch. So they don't actually see this. They actually see a pitch simulated at moving at, whatever, like 90 miles an hour or 80 miles an hour if it's a curveball, and it breaks a certain way, and then they have to respond as fast as possible. Did this pitch trajectory match what they were looking for, okay? And we did this with the Columbia baseball team. Surprisingly, they don't stink. They're actually a very good baseball team. I know this year the football team was good too. So, and so we did this whole experiment, right? But at the end, what we did is we did resting state. Okay, so the notion was, all right, you do this task, and we've analyzed all this data, but now we collect resting state. We said, well, is there anything different between the brains of the players and the brains of the non-players in terms of their resting state dynamics? And just to go right to the punchline, what we found, so in this case, instead of having events where we would actually use this machine learning to do classification or what have you, we isolated from the event-related part of the experiment areas, particularly in supplementary motor area, that correlated with performance. And we looked at the power of alpha and beta oscillations in the EEG and used those to look at functional connectivity across the brain, so basically between this SMA and other areas. And without going through all the details here, if you look at what connects to what and the group differences, what you see is that this is all consistent with what one sees in the literature of some kind of motor learning happening in the heads of the baseball players relative to what's happening in the heads of the matched control. So these are just grad students that don't play baseball, but the same age and same gender. And so this suggests that, what is resting state, right? We always want resting. Is the brain really at rest? Well, here there's been some discussion about, well, post-task resting state is actually a thing in where you're evaluating how you did in the task, and maybe you're learning from it, updating models. This suggests that that's happening. So in addition to, as you're doing a task, you're potentially updating on the fly based on your certainty, you can also be learning here, after the task in resting state. And it seems to be something where, for the experts, since they kind of value their performance on the task, they were all much more excited. They wanted to know, all right, how well did I do after then the grad students? They just cared about getting their stipend. So this is also another interesting thing that we're continuing to pursue in terms of some of the nuances of this. Anyway, so I want to end by just saying, all right, so I haven't talked about anything clinically, and one of the reasons I'm here is that I'm super excited about kind of working with many of you, trying to use some of these methodologies to really help people with mental illness and potentially better understand facets of psychiatric disease. And so I have a number of different projects that I've been working on up here with some of you. I just wanted to quickly make a couple of shout outs to that. So one effort, we've been doing some work with Samir Sheth, who unfortunately is left, and Yagna Pagnath, on using simultaneous EEG fMRI to look at cognitive control in OCD. And this is actually just some of our preliminary data looking at differences in the spatiotemporal dynamics using the same methods I just talked about, where we see differences between OCD and match control that include parts of insula and also anterior cingulate, and how we're relating that to behavioral performance. So once again, this is actually an experiment using the multisensory interference task, classically used in OCD patients, and we're getting a better idea both of differences in the activity, and then Samir's kind of long-term goal is to say, well, can we learn anything about better targeting of neurostimulation by combining space and time measurements together with behavior. Another piece of work with John Mann and Noam Schneck, where so far we've only used fMRI by itself, but as we move forward, we're going to use the fMRI EEG methodology I talked about, was to be able to track spatiotemporal networks underlying cognitive processes associated with complicated grief. And so this is just some work that was recently published from fMRI in Biological Psychiatry, CNNI, where we show that we can track activity in kind of mind-wandering states that relate to different measures, clinical measures, such as avoidance measures. And this is across different individuals. And so the idea is, are there kind of neural markers that we can tie to clinical markers and behavioral data that give us a better picture of like coping processes in things like complicated grief? And lastly, a project I'm super psyched about. In fact, I want to figure out how to bring it here, right? Because right now we're doing a clinical trial at Medical University of South Carolina. I'm an engineer, so if I can put two things together, why not put three? So I put EEG, fMRI, and TMS all together, right? And in fact, this is our system here at MUSC. And the idea is to create a closed-loop system for precision delivery of stimulation. And our clinical trial is focusing on drug-resistant major depressive disorder. And so the idea is that, TMS for treating depression has, limited efficacy. It's about, I think, about 30% of individuals it works on in a reasonable way. And part of the hypothesis is that, well, what matters is when you deliver the stimulation relative to, endogenous rhythms in the brain. We're hitting like, dorsolateral prefrontal cortex, but we're really, the hypothesis is that the target is really somewhere here in the cingulate. And so maybe that functional connectivity between DLPFC and rostral ACC is dependent on the ongoing alpha phase in prefrontal cortex. So these are just preliminary results on controls, but what we find is this is actually the phase of the individual alpha frequency across populations just normalized. This is what the bold response we see in rostral ACC as a function if we deliver the TMS pulse at that time. And we can see that there's a significant difference between if we hit at the bottom phase of alpha versus at the high phase or rising phase of alpha. So there's clearly variation in the activity we can see in the bold based on the timing we're driving the stimulus, the neurostimulation based on the EEG. So this is just another thing I'm trying to work with Randy Auerbach, who just arrived not too long ago. So, and I'm open for many more collaborations. So with that, I'll just acknowledge my collaborators, of course, the postdocs and students that did all this work, support, and also any more information from my lab website, or I'm also the chair of the IEEE, so this is the big electrical engineering society in the world. I'm chair of that BRAIN initiative, and part of our goal is to take engineering and the expertise engineers have and kind of try to work more with clinicians and understanding how to improve mental health and well-being. So I'll take any questions. Thank you.
Paul, thanks very much for really a fascinating talk. I think I recognize some of the research that a guy named Peter Venkman was doing at Columbia around the time that Ghostbusters was released. So just to start the discussion, I'm going to ask a couple of questions. I assume there's inherent limitations just in terms of the signal strength of bold fMRI and EEG with scalp electrodes. So those are just limiting factors that you have to deal with. Is there a phenomenon that's potentially a problem, like discontinuities between those? In other words, when you see some bold activation of a specific voxel that doesn't correspond to EEG, how do you deal with that?
No, that's a good question. So you can only capture kind of the shared variation, right? So one of the key things is when we look at this analysis, what we care about is what can I explain by EEG only, what can I explain by fMRI only, and what can I explain by both, right? So we don't want to throw out anything that might be explainable just using one modality. And the reason I focused on what we see in the joint space here is to show that there's value in the joint space. But really, when we think about this as a modality, we can do the same things we wanted to before with classic fMRI, classic EEG. We can just do more by adding in the joint analysis.
Questions?
The hippocampus is famous for generating replay and driving replay in cortex as well, and I'm wondering if you have any information about whether it's replaying in the same areas of cortex that the anterior cingulate might be in your case?
Yeah.
And if there's overlap there?
It's a really good question. I mean, the problem is, so with EEG, scalp EEG, you really can't get hippocampal activity. It's just too deep, right? And so, one thing would be, could we do an experiment where we also had some form of depth electrodes in there? That's always tricky, also going in a scanner. So I think the only thing that we could do is, and in fact, I've been talking to Josh Jacobs here, who does some of this work, is we could try the same experiment, right, on individuals that have, ECOG and depth electrodes undergoing epilepsy procedure, and just look at the timing of some of these replay and see if we see replay at the same time in hippocampus that we seem to be seeing triggered by this ACC. But it's a good question. And in fact, I'm also a believer that this notion of replay, I think, is a fundamental because it's key for learning, right? And one of the things, once again, I also came from a bit of a machine learning background. The problem with machine learning, 20 years ago, where they still had neural networks and all of this, is we didn't have enough compute power and examples to train on. And now almost all of the really big leaps are, well, I have more CPU cycles to train and more data to train. So I think sometimes we might take for granted how much we're adapting and when we're adapting, and there's multiple ways we're adapting. So I wouldn't be surprised if all of the different ways of replay are somewhat can be distinct from one another.
Bill?
Thank you, Paul. That was fascinating. To follow up on the previous question, we don't necessarily have to tap into the hippocampus to look for some evidence of this replay. So I'm, as you clearly showed there, where we could look at the different brain areas that seem to be lighting up sequentially. And during sleep, which is another whole time when we're doing this replay, and as you and I were talking about earlier, we've both managed to follow sleep pretty easily in magnets. There are ways that you could kind of prompt the first stimulus that's part of something that you just learned, some task you just learned, and then without tapping into the hippocampus, start looking at these same sequences of activity that tells us a little bit more about reactivation, or when reactivation isn't working, when the memory process isn't doing what it's supposed to be doing. So there are ways to get at it, I'm assuming, without having to get right down to the hippocampus, right?
Right, and sleep, I'm glad you brought that up. I mean, sleep is one of the, you know, the most well-known times in which replay seems to occur. I think that what I would love to do is come up with an experiment where we could do that and then behaviorally test, in fact, was that replay used for learning? So we find, let's say, these patterns, they're expressed, and then we see our specific patterns associated with particular recall of something that we can look at behaviorally. That'd be great idea.
David, then Blair.
So I have a question about timing. I understand in many sports like downhill ski racing, gymnastics, ballet dancing, even conductors with music, they pre-play or replay something that they want to practice in imagination. I mean, they'll run the course once in the Olympics, then they'll remember it and they'll go over and over and over. My question is, when they do that, do they have to do it at the same time rate, or can the rate of replay be variable?
That's a great question. I don't know the answer. This would seem to suggest that the replays are faster. That of course it's, you know, we in the one of the problems with this data that I showed is, though I show that it's correlated with confidence, I never show that there's anything with learning. But my guess is that they can probably compress time in a way that they can kind of practice in their brains through replay at faster rates. I don't know, but that would be my guess too.
Blair?
Hi, Paul. So I have a local, I'm here.
Hi.
I have a local comment and then a global comment. The local comment is, yes, it is a shame that Samir is leaving Columbia and has gone to Texas, but the OCD patients are still here in my clinic. And so if you have a graduate student who wants to pursue that work, I'm very happy to talk with you about it. But more globally...
Well, also, we're in the midst of a recruitment, right?
That's exactly right. But more globally, it's exactly what I was going to, is that the department is really trying to work with neurosurgery and neurology around a neuromodulation initiative, and you obviously are a very important part, and I have someone who maybe it would be really great for you to speak with. But the third one is our department is actually creating a new EEG core resource. And I know that you're connected with Randy, but Bill is obviously part of that, and Dan Javid is part of that, and Jürgen Kaiser is part of that. And it's something that actually we hope to really engage you in more broadly, so that some of these new tools for looking at the brain can actually come to more of our clinical populations. Because for us as psychiatrists, the key now is how do we find targets in the brain that we can actually use to test novel interventions as well as understand mechanism, and having better and more precise targets than what we have with just bold activity is really a goal for our future. So we look forward and really want to partner with you.
Great.
Paul, let's take this question here.
Hi. So my question is a little bit of a methodological question. So when you look at the signal in the EEG and look at the variation in the voxels based on the hemodynamic response, basically we are regressing the voxel activity with the EEG activity, right? But they are on a completely different time scale. So we need a lot of variance to be able to capture or get enough weights. So how do we optimize that? Do we?
So great question. So I didn't show any of the math. One of the things that we're doing, right, in these encoding models is we have to, one, like you said, their variance is such that we have to kind of localize the meaningful variance. So there's a whole SVD decomposition that we have to do in order to kind of, collapse that variance that matters into fewer dimensions. We also do a regularization via ridge regression, and a couple of other tricks, right, to be able to get a model fit. I think the point also is that with these types of encoding models, you get a model and then you get to see how good is it, right? So there's the goodness of fit you can do by saying, all right, how well do I predict the bold variation from the EEG variability on unseen data and things like that. So you get, you can say it's either a good model or a bad model. And so I think that that's kind of also an element to this. We're clearly not, there's so much variation also that's, like Jeffrey was saying, that's likely to not overlap or co-vary, right? And there's nothing we can do about it. And that's okay. We have to invent some new other type of imaging. But what I think we're able to show with some of this is the fact that there is meaningful information, particularly on about how activity kind of, the dynamics start to evolve, even within relatively short time frames with respect to the bold. Now, I think also what's really exciting, and I didn't talk about at all, is bringing in other modalities, constraints, prior information. So one thing is we've been looking more closely now to use DTI, DWI as a way to really constrain connectivity and interaction models, use that also kind of as a regularization for fitting these models, as well as other types of physiological information. One thing that I'm also very interested in is looking at, particularly with these visual tasks and decision-making tasks, pupil dilation, how the eyes move, to get indexes of arousal and see how those co-vary with these models. So I think that ultimately there's going to be this multifaceted approach of measuring different facets of brain activity and physiological response to make inferences about, what's going on in the healthy and diseased brain.
Bran, is Howard here?
No, he's with his mother getting cardioversion.
That's an overshare. Okay. So what you get in response to your task is a decision or some kind of choice that's made. And embedded in that are possibly multiple sources of, cognitive analysis or bias and so forth and so on. I mean, it could be there's a certain cognitive speed, like with Wall Street traders, there's now this like hyper fast computer system that can, some people's cognitive acuity may be similar. On the other hand, you may have people who are impulsive and they act on impulse. So how do you just deconstruct that?
Yeah, I think ultimately it's going to be these types of measurements have to be informed by then kind of cognitive models of processes, and then those models being used to then create experiments that test the model with the new data, right? So I see this as a way to measure spatiotemporal dynamics and get at certain latent states we couldn't measure before, but it's only as valuable as kind of the models and hypotheses that they generate and we can test. One thing I can tell you about is, so we've been doing more and more work. This actually isn't fusing EEG and fMRI, but using fusion of pupillometry, electrodermal activity, EEG, to look at tracking arousal changes in individuals, particularly as they do a task in VR. So we use virtual reality a lot because it engages the subject. And the ability for those signals to generate neurofeedback that allow individuals to regulate their own arousal levels so that their performance in a task improves. So what we want to do now is take a model of how we think that arousal circuit is being modulated and put it in that feedback loop, and using kind of classical engineering techniques, identify that system, okay? So I think, one of the exciting things I see, so just as a scientist, forget about the fact that, helping individuals with mental illness is, I think, a very important thing. But when we have groups of individuals that have certain dysfunction, that we can potentially look at differences with another group, kind of like the baseball player group, then I think we're even going to get more insight into what should go into these models.
I'm wondering, Deborah and Susan, so you mentioned that cognitive hypotheses need to be formulated too. I'm wondering how, the others, kind of a model or system that's applied to, behavior and psychopathology, psychodynamics would be sort of, would approach this. Have you?
Well, one thing that comes to mind for me is there's a system for classifying what a person's specific conflict-laden words are, and they've shown that they can be more or less accurate identifying them depending on the length of time they're shown for. And there are some original evoked potential data from Chevron and Bond in Michigan about sort of descending cortical input in response to conflict-laden words. And I wonder if you could build a nice model of defensive operations, like where you could actually, show sort of the rising stimulus in the brain of the conflict-laden word and some attempt to shut that out of awareness using this model. So I think that would be a really terrific little project. I've been wishing we could find a way to sort of take a look at that study again, because, defensive operations are a big part of what we do. Like, how do people protect themselves psychically from sort of knowing what they don't want to know?
Right.
So that would be interesting, I think, something along those lines.
So it's like, your work wasn't hard enough as it is with the limitations technically and so forth and so on. So cognitive neuroscience is the kind of a theoretical framework that you can use to, begin to sort of deconstruct the data that you get. We're going to make it harder by introducing the idea of psychoanalytic theory, for which there's limited empirical basis for, but yet at the same time has a lot of face validity and is historical utilization by the psychiatric profession.
Well, I mean, I think, so the work that I've been doing with John Mann and Noam Schneck, I mean, I think I've learned a tremendous amount in terms of, how you can take, we're always looking to relate measurements to something, so like response time. But if there are these measures that obviously in psychoanalytics have been really used for long periods of time, there's basis for them, we can get better ideas of how some of this information is expressed across those. And that allows some of these models to get built. And so, I'm super excited learning more and more about psychoanalytics and psychiatry with interactions with you folks.
Well, there's a pretty long-standing system developed at UPenn by Lester Laborsky for classifying patient stories and dreams and things in terms of conflict-laden words and material that has been used a lot in psychotherapy research and is pretty interesting. So,
Bill.
Sorry for this low-level question, but I was interested. What you're presenting to us so far is mostly the brain in action. That's what fMRI is really about, tasks and decisions. I wonder if it also would have a use in just diagnosis of, for example, seizure foci or advanced diseases where you could use a kind of a stereotactic view of what each one of these modalities will do and putting them together to refine diagnosis at rest.
Yeah, no, I think that's a great question. In fact, Kathy Chavon just sent me like an email yesterday or so talking about like a project that she's working on with regard to this. I think, yeah, it's kind of interesting because in some sense, I don't even know the problem space that we could potentially employ this as a methodology. And part of it is to see what that problem space is, both for kind of basic science, but also for clinical science. And I think that, we've done some as part of different projects here. For instance, one with Jeff Miller, where we were recruiting as part of his R21, where he was looking at opioid receptors, kappa opioid receptors, and trying to look at correlates between expression of that in PET and some of these markers. And in fact, so I think it's a matter of getting NIH to pay for it, and then we can work together and do it.
Well, listen, Paul, that was a great presentation and discussion, and I think, as I said before, in the words of Claude Rains at the end of Casablanca, this is the start of a beautiful friendship. So we'll have you back soon. Thanks.
Thanks, Jeffrey.